go. So listening to the brain for concussion care. I'm Nina Kraus, and we have a lab here we call Brain Volts. And I'm going to start with the punchline. And the punchline, this is very important, especially coming out of the previous talk. The punchline is we have discovered a biological marker for concussion that is objective and tracks. It identifies 90% of concussions, so it has a very high sensitivity, and it clears 95% of people without concussion controls. So I am going to make the case that this is going to be a very important addition to the various tests, and also the punchline is that, well, we know that there's not going to be a pregnancy test for concussion. It's going to be a combination of different tests, like the ones, the balanced ones that we just spoke about. But this is, I think, really giving us some unprecedented, needed biological information. So why? Why would we have this biological marker that involves the auditory system? And I'm going to give you some background, maybe more background, and since we have folks from Northwestern here, I'm talking also about my personal history here at Northwestern, but specifically why sound? So sound is a tremendously powerful force that is central to human communication. One of the aspects of sound is that it is invisible. And like many important forces in our lives, like electricity and magnetism, which are invisible, sound is invisible. So in contrast, if you have a seen object, it is, it is very easy to identify with a number of different attributes. So you can identify the ball. It has a certain shape, a certain feel, a certain color. It has aspects that are materialistic, right? Vision is a materialistic sense. As opposed to, with sound, you have these attributes that are much more abstract. They're abstract, they're fleeting. Sound, by definition, moves. So it doesn't stay in one place, like this thing here that we can look at. And did I say it was invisible? So I also want you to contrast auditory and visual processing, because one of the points that I'm going to make is that the reason that our biological measure of concussion, our marker of concussion, involves the auditory system makes really good sense, because the auditory system requires that we do some of the most computationally complex processing that we ask our brain to do. And so it is not surprising that if you get hit in the head, you will disrupt this very, very delicate machinery. So one of the things that, there's nobody sitting here. Come sit up here. So one of the things that we can look at is neuronal speed. So if you stimulate with sound, you see that neurons fire very, very quickly. So they fire, this is just in a millisecond's worth of time. As opposed to with visual information, so the light falls on the photons, and then eventually you see this action potential, or the activity of neurons happening. And this is something that is happening more than 40 milliseconds in time compared to 1 millisecond. And so I love signals, and I love animations. So I want you to just take a look at this for a few minutes. Look at how quickly the sound information is occurring and how much longer it takes for visual information to pass down the neurons. Okay? So that's one thing. But now, within the brain, the processing speed is even faster. It's faster still. We're even talking about less than a millisecond. We're talking about microseconds because, when we even figure out one of the ways that we make sense of sound is the brain has to compute the time that it takes the sound to hit one ear and then the other ear. And what we're then asking our brain to do is do some math and figure out what is the coincidence. There are these coincidence detectors in the brain that will compute the amount of time that it took the sound wave that's happening over here to reach this ear compared to that ear. So, have a look at this. Sound is coming into, say, the right ear, and then the left ear will be hit, and at a certain point, you will have this whole cascade of neurons in the auditory brain that are sensitive to this microsecond processing. Okay? I want to tell you just a little bit about me, and one of the reasons is that this is Northwestern, and a good deal of my history involves the wonderful people who I have interacted with here at Northwestern. So, beginning a long, long time ago was my mommy. So my mommy was an immigrant. And I lived in a household where more than one language was spoken. So sound, important things were happening in sound. Importantly, too, she was a musician. She was a pianist. And one of my favorite places to play was underneath the piano. I would just take all my stuff and go sit under the piano and be there. So, sound. I'm a biologist. And the idea, personally, and the idea of our lab of BrainVolt is to, we study sound processing, making sense of sound biologically, but importantly, we do it to inform health, to inform human health. We are interested in how the discoveries that we make in the laboratory can be applied. That's absolutely critical to our mission. And so I've been chipping away at this for a while. Back to Northwestern, I began years and years ago. Anybody recognize this guy? Well, his name is there. But anybody recognize him anyway? This is, Raymond Carhart, who was the founder of audiology. And I met him many years ago, and he almost took me by the hand. And after listening to what it was that I was interested in, and he introduced me to this guy, Peter Dallas, who again was a very important force at Northwestern in auditory health and auditory neuroscience. So I began with him counting hair cells in the cochlea and measuring neural activity in the 8th nerve of the chinchilla, something called two-tone suppression of the auditory nerve. And this is where my mommy comes in again, because at a certain point she said to me, Nina, what are you doing? and I realized that there are maybe 16 people in the world who I could talk to about two-tone suppression in the deep way that I was thinking about. And I realized if I couldn't talk to my mom about what it was that I was spending a lot of time on, I didn't want to be doing it. So I started sticking needles in bunny rabbit brains and looking at recording from single neurons and seeing how the response to the neuron changed once the sound meant something to the animal. Okay? So, you start out measuring the response to sound, and once the sound has meaning, the brain response changes, and we can measure that very, very precisely. But again, interested in humans. So what I've been wanting to do, and I've worked on for a long time, is searching for the kind of granularity that we can have in animal models. We still have animal models in our lab, but we are interested in humans. So, how can you measure the kind of electrical activity with the granularity that you can in animals? Well, we can. Okay, so there's really good news here. So I've been searching for this granularity of sound processing and also searching for metrics that measure individual differences, because we are interested in individuals, and an individual's life in sound certainly influences how his brain responds to sound, and has all kinds of implications for communication. So this is me, very happy. Actually got a ticket for this, for swinging across Ashland Street. But very happy in that we discovered the frequency following response is a biological response to sound that has tremendous granularity, and it gives us a lot of information. I need to tell you a little bit about this before I come into concussion. So even though we are interested in big health issues like concussion, language impairment, what happens to our ability to make sense of sound as we get older, we're basically signal geeks. We love signals. So sound waves, which this is the movement of air molecules, and brain waves, electricity. These are signals. These are very, very concrete, measurable things. So what can we do here? We can deliver a sound wave and measure the brain's response to sound. So the scalp electrodes just measure the electricity in response to sound. And what you can see here is, if you look at these two responses, or these two waves, this is a sound wave, this is a brain wave, what do you notice?
Pretty similar.
They're kind of similar. Thank you. And they're so similar that you can take the brain wave, play it back out through a speaker. Just like I take an electrical signal from my electric guitar and feed it through my amplifier, so you can sonify it. You can sonify the electrical activity that you measure from the brain. So, here is the sound wave.
Dot.
And here's the brain wave.
Dot.
Okay. Some more examples.
Dot. Dot.
A scale and the brain's response to a scale. Little Mozart, and then the brain takes over. And my favorite. Okay, so the point is we have a lot to work with, and so from a single response that I can measure from anybody in here's brain to a speech sound, to a complicated sound that has a lot of sound ingredients in it, we can figure out how good a job an individual brain does at measuring these particular components of sound processing. And importantly, and I use a mixing board analogy here, because if you have expertise, it's not as though everything gets better, or if you have a disorder, for example, like concussion, it's not as though all aspects of sound processing are affected. There are only very, very specific aspects of sound processing. So it tells you biologically, first, A of all, is there a bottleneck? Is there a problem in how the brain processes sound? And two, what is the nature of that problem? I can tell you that the signature, the neural signature for autism is very different from the neural signature for reading disorder. And both are language disorders involving sound, and we can see that. So we can look at what is the strength of the fundamental frequency. So I'm really just talking about some of these components because they're going to come up. The strength of the fundamental frequency is very important for pitch cues, and it's very important for identifying objects. The harmonics are important for distinguishing speech sounds and distinguishing instruments. We look at timing of these. Again, this is microsecond timing in quiet and in noise. And we can see how accurate the response is from trial to trial by actually correlating the stimulus with the response because they are so similar. So we can have a measure of accuracy, and we can also look at stability, how stable the response is from trial to trial. Importantly, this is back to individuals. Individuals have their own signatures. Here are two individuals, and 4 years later, here are the same guy's responses. So even though there are some subtle differences that are interesting, you notice that there really is a signature for every single one of us. And that's very cool. So practical advantages of the FFR is that it's objective. So certainly for concussion or for any kind of evaluating anyone with a language or communication disorder, having an objective measure is obviously important and easy. It's vetted, very well vetted. It's mobile. You can take this technology around. It's fast to do. It's repeatable. And it is, you can come up with a uniform measure that you can use in a baby, in an older person, for a variety of different disorders. Okay, finally getting to concussion. So processing sound is one of the hardest jobs that we ask our brain to do, especially because of this tremendous timing and the interaction of processing of all these different elements of sound in the brain. So, getting hit in the head, it makes sense that getting hit in the head would disrupt this delicate machinery. And that was the hypothesis that we went in looking at for concussion. So do you think that this is a plausible hypothesis? Given what I've told you about sound processing in the brain, given the granularity with which we can measure sound processing, does it make sense that if you get hit in the head, perhaps this very delicate, intricate processing machinery can be affected? With me?
Yes.
Okay. So, so concussion, we have a number of phenomenal partners, and BrainVolts is our lab, and we do what we do, but we could not do the work on concussion if we did not have two amazing partners and two amazing group of participants that we have been working with. So one is Cynthia Labella, who's a pediatrician, who is the director of sports clinic at Lurie, and she is the one that has been our partner in looking at school-age kids, about an average of 13 years of age. And then there's Tori Lindley, who is here with Northwestern Athletics. And he is the head trainer. He is the person who oversees all of the sports. And we have been working with him. He's very, very active in concussion care management and student physical health. And we have been working with the football players here. So a lot of this was covered in the previous lecture, the fact that concussion is an issue. We know that at least there are 2 million concussions in the United States annually, and that's reported concussions. So it's certainly a big problem that has cognitive, emotional, academic, and neurologic consequences. And there are a number of challenges in concussion management. And again, these really were covered beautifully. You can understand why from the previous talk that the symptoms can be nonspecific, they can be subjective, and that the recovery, what happens so often is that the current measures aren't good enough in that the people have they're given a clean bill of health. Your physical exam is normal, your MRI is normal, and yet people continue to have these symptoms that are troublesome. And so, this leads to uncertainty and frustration, which is why we very much need neurobiological markers. So, biological indices of concussion, again, this was covered previously. There have been publications talking about different white and gray matter changes occurring with concussion, but again, this is on group levels. There have been a number of electrophysiologic studies in all the modalities. And again, the data have been conflicting. And what has been often lacking is individual specificity. There have been blood markers. Again, you heard about the pathways involved there and the limitations in the blood work. And there are genetic predispositions, there are genetic makeups of an individual that will influence the kind of outcome that you might expect and the kind of difficulty that they might have with concussion. So why the auditory system? Well, I spent about 20 minutes talking about why the auditory system, and so that's why. But the idea is grass is rustling, and making sense of sound, again, is one of the most complex things that we ask our brain to do. And we really need, it involves our cognitive, our sensory, our motor, and our reward systems. So it really involves so many networks of the brain, which is why auditory processing is actually central to so many language and learning and communication problems in general. So with blast injuries, what you can often see is frank hearing loss. So here is an individual who has a change in their hearing threshold. But there are other people who have normal hearing thresholds, but they have difficulty understanding speech, making sense of sound. So clearly there is something in the brain that is not computing. And I think you'll be hearing about some central processing tests that people have shown that a consequence of concussion is difficulty in various aspects of central auditory processing. So what do we know? In head injury, mild and moderate TBI, there have been a number of publications that have indicated that there is difficulty making sense of sound, and especially making sense of sound in complicated backgrounds, so hearing a noise. There have been long-term effects. So again, other symptoms resolve, but the difficulty hearing a noise persists. So the literature is pretty, there's converging evidence that hearing, speech, and noise is an issue. And sports-related. So there really is just a single study that we know of that has specifically looked at auditory processing in sports-related injuries. And what we see here is in this study, and it's a small study, 8 individuals either with high-impact sports or lower-impact sports, and they were given the typical auditory processing behavioral tests of thresholds, speech perception and quiet, tone pattern recognition, monaural separation and closure, and dichotic listening tests. So speech and noise wasn't directly measured, but in the low impact sports, none of the people showed any of these central auditory problems, whereas 5 out of the 8 in the concussed, high impact sports did. And also of these 5, the deficits were greater than 2 standard deviations. So you're really talking about severe deficits. They're not just right on the threshold here. So the question here is, what about hearing and noise? And I'm going to jump ahead This is some of our data. I'm going to be telling you about 2 studies. One that was done with the kids at Lurie, and these are basically 13-year-olds, and that's what you're going to be seeing here. And then the data that we have been gathering with the football players here at Northwestern. So in the the children who are seen at this this is a clinic where the symptoms are this is they continue to have symptoms, so they're being seen by this sports medicine clinic. And what we can see is that if we look at hearing and noise, the 50th percentile is right here, and our participants, most of them are scoring pretty low on that. And this hearing and noise work, I want to point out Ellie Thompson, who's sitting right here, who's been leading the hearing and noise part of this, and also a lot of the other work as well, has benefited from her participation. But other things that she has just gotten us thinking about as recently as yesterday is the fact that when you have hearing and noise measures, you can have a signal here, and this guy is trying to listen to the signal, and the noise can be in the same location as the signal, or the kid back here might be making noise, and so they can be separated, the noise and the signal. And what we seem to be seeing is that for both situations, both the control and the concussion kids, you can see that the kids who have had concussions have poor hearing and noise in both conditions. And what's important is that the control kids they've been recruited from the same clinic. Okay, so they have come into the sports clinic, the sports medicine clinic, with a knee injury or with some other kind of injury. So they have been matched in every possible way that we can think of matching them. And again, this is largely Ellie's work in collecting these data. So we have this connection between concussion and hearing and noise from work in the literature and from our own work. What about the connection between hearing and noise and the frequency following response? Well, it turns out in a number of studies, if we look at particular aspects of sound processing, so I want you to focus to think about the fundamental frequency, which is a very important pitch cue, so that will help me know if that's Sue speaking, or is it Ellie, or is it Frank? So it helps us identifying sounds. Okay, so knowing what to leave out and what to pay attention to. And what we see is that the frequency following response tracks very well with hearing and noise ability. And I want to, I think you're probably all familiar with these tests, but the kind of hearing and noise tests that we typically use is see if you can hear a sentence in here. Hear a sentence? Try this one. Get it? Okay, so this is the kind of test, when I'm talking about hearing and noise tests, that we can measure in the lab. Okay? Okay, and so there's this relationship between the strength of the fundamental frequency and hearing and noise ability, and we see this across the lifespan. There's another aspect of the frequency following response. Remember, I said that the stimulus and the response are so similar that a stimulus to response correlation is a meaningful measure. And so we can correlate the stimulus with and the response and just get a number. I mean, it's just an R value. And we can see that there's a relationship between hearing and noise, and we call it the accuracy with which the stimulus and the response are linked. And finally, we've been looking at timing, certain aspects of timing, especially timing of that are important in consonants, distinguishing one consonant from another. And again, we can see a nice relationship between hearing and noise and timing. So we have now this relationship between concussion and hearing and noise. We have a relationship between hearing and noise and the frequency following response and certain components. What about the relationship to concussion? Drum roll. So this is one of the studies that we have published. And again, I want to point out, this is Cynthia Labella and Catherine Cook, who works with Cynthia. Ellie, who I've already mentioned, who really captured all of the data points that we are talking about here. Jen Krisman and Travis Weitschwach, and they're all part of BrainVolts, and everybody is part of the Northwestern network. And this was published in Nature, Scientific Reports. If you want to have a good, the Washington Post did a really nice write-up of this piece. So if you Google my name and Washington Post and concussion, you'll find it, and they do a really good job, as these guys do, always really struggling to make the science, why an auditory test, right? I mean, it took me 20 minutes to set that up, and they do it in, half a page. Anyway, so we have this concussion group of kids averaging the age of 13 at Lurie, and then we have a control group. Okay, and so what did we find? So we looked at the strength of the fundamental frequency. That's one of the factors, and this is from our norms, and I don't see my norms up here. Anyway, this is from published norms of the frequency following response, and what you can see is this is the fundamental frequency in the controls, and notice how the kids in the concussion group have a smaller fundamental frequency. Okay, so that happened. That's one of the frequency following response measures. Then we looked at the stimulus to response correlation. Okay, how well was the response reflecting some of the stimulus characteristics? So this is something that, again, we saw that the stimulus to response correlation was lower in the concussion compared to the control group. Okay, so these are now two measures. One is the fundamental frequency, the other is the stimulus to response correlation. So something that gives you pitch information, something that gives you accuracy information, and then we looked at timing. So this is timing to speech sounds, and again, if you look at a brainwave to speech sounds, you get these nice electrical patterns. And so the concussion kids are in red, and we noticed that there are some delays, and importantly, not every wave is delayed, so it's not as though it's inherited. And by the way, these all had normal auditory brainstem responses to clicks. So there was no latency difference in these groups to click sounds. A click is just an onset response, and a speech sound has so much language-rich information. So we saw that. So taken together, we can look at these measures of strength of the fundamental frequency. So we can think of strength, we can think of pitch, we can think of speed of processing in terms of looking at neural timing, and we can look at accuracy by looking at this measure of stimulus to response correlation. So these factors all together are important, and importantly too is like some of the other factors that we looked at are not at all tied with concussion. Okay, so again, I think it's very, 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 very important to be thinking about with this measure that it's, yes, A of all, is there a problem, a biological problem in auditory processing? Check. But it's not just how big is it, and it's not something that is affecting every single aspect of auditory processing. It is very, very specific. There is a specific concussion signature, which brings me to where I started out, which is using, doing a binary regression and modeling the data so that we could look at, if we look at these three components together, how good are they at identifying whether or not a concussion happened. And we see this 90% identifying concussions when they're there, and 95% clearing individuals who do not have a concussion. Importantly, too, this measure of the frequency following response, this measure of making sense of sound, which involves, it's a snapshot of auditory processing that involves bottom-up and top-down processes. So it is this very rich measure of sound processing, and it tracks with symptom severity, again, with the kinds of behavioral symptom tests that we heard about in the previous talk. What about recovery? This is a really, really, really big issue. We don't want to be sending athletes back to play when their brain isn't ready. And a lot of the typical clinical measures will clear people to play, both the behavioral measures and the biological measures in the physical exam. So we were able to follow, out of the 20, 11 of the kids with roughly a month interval in the test-retest. And so, what did we see? So again, it's interesting that the particular aspect of sound processing that lingered, but that also got better is, so here is the first visit, clearly below the norms, and the second visit, which is now coming into the normal range. And these are group data. Of course, it's very important to look at individuals. So, we're going to just give you an example of the kind of thing that we have seen in individual players. So, I'm getting ahead of myself a little bit, as I want you to think about the, we're looking at recovery not only in these 13-year-old kids in the concussion clinic, but also in our student athletes here at Northwestern. And with football, we're seeing roughly one concussion a week. But you might see something like this where you have a baseline response, concussion, and you can see that the FFR is diminished in some way. And he might be clear to play, but again, you understand because of the biological mechanisms that were spoken about in the first talk, the brain can continue to swell, it can continue to have damage long after the behavioral symptoms are clearing into play. So at the moment that this is just an example. And then 2 weeks later, which just happens to be our test-retest interval, he's fine. And so how important would it be to wait until this happens to send an athlete back on the field? Because at this point, he's probably the most vulnerable, or one of the very most vulnerable times during his post-concussion history. You can have the season continue, and as we've also seen, some athletes have gotten a second concussion and still symptomatic, cleared to play. Still, we can see that the FFR is showing some abnormality, and 2 weeks later, the symptoms resolve. So, this needs to be validated. This is just to get you thinking. We need to be looking at, concussion, acute stage concussion, and recovery very, very, very carefully in large studies, and this is something that we're scaling up to do. But this is just the kind of thing that, if we could have this kind of information, it would be immensely informative. So this is our first paper with the Northwestern athletics folks. And I had already mentioned Tori Lindley, Danielle Colgrove, here's Ellie again, and Sebastian Odemeyer, who is right over there, Jen Krisman and Travis Whiteschwach, and also Sylvia Bonacina, who's sitting here, who is now involved in our ongoing work with especially football players. So this was published, or is in press, in Neuroscience Letters. So it should be out, I think, in a week or so electronically. But here's what we found. So we tested all 105 football players at preseason. We tested them so this was last July. And simply what we did is we divided them based on whether or not they had had a single concussion. And 25 of those 100 athletes had already a history of a previous concussion. And so what we did is, within this group of 100, we very carefully matched them, and matched them in every way that we could with another 25 who were on the same team. Okay? And what we could see is that there is a difference, and again, in this fundamental frequency, there is a difference in the kids who did not have a concussion and the kids who have a history of a previous concussion. Athletes, I call them kids, who were cleared to play. I mean, these were all functioning, working, playing members of the football team. Also important to know is that if we look just at the football players who had no prior history of concussion, but if they're on the football team here at Northwestern, they have had a history of playing football and getting knocked around. So this idea of subconcussive injury, it's kind of interesting to us that the athletes who had no history of a frank concussion are still showing us frequency following responses that are lower than the 50th percentile. And of course, the ones with concussion are lower still. Okay? So we really have something to work with here. So I'm going to just sum things up. We have this biological marker that is very sensitive and specific to concussion, different from control. We have this measure that tracks with concussion severity, so symptoms. We have a measure that tracks with recovery. And so, we can think about having a biological marker. We see that concussion kids who have had concussions have slower and smaller neural responses to speech sounds, and very specifically, there is this disruption of the processing of the fundamental frequency, the timing, and the accuracy of the response. As concussion symptoms abate, aspects of neural processing recover. There's high predictive power in the FFR, as I've shown you. And so this tells us that concussion diagnosis and management, recovery management, can be strengthened by tracking changes in the auditory system. I think that we're able to bypass some of the limitations of existing tests. And again, I am not suggesting, as I said at the beginning, there is likely not going to be a single test, a single pregnancy test for concussion, but rather it will be a panel of very fine-grained measures. And this is one of the finest that I think is around. And so I think that it really is a very promising biological marker of concussion. So, what next? We need to have very robust and need knowledge about these outcome measures. So, yes, we want a biological marker. We want it to be standardized. We want it to be biological. We want it to be objective. We want it to be personalized. I want to go on record as saying that I think that playing sports and being physically active is one of the very best things we can do for our health, for our children's health, for our nation's health. I identify as an athlete. So I'm tremendously interested in health and in managing sports injuries, because we want to be able to do the very, as biologists, we have all this information now that we can bring to bear to help people be as healthy as they can possibly be while they are playing these very, very important sports and they are involved in all kinds of physical activity. So, we're needing to scale the work. We have this phenomenal infrastructure in place with Lurie Hospital and with our Northwestern Athletics, where we can look at other teams as well. And we really want to capitalize. What I've shown you is very, very strong proof of concept. These are small studies. They need to be scaled to larger studies. And I'm telling you where we are sitting today as a lab. We have been writing grants constantly. Our Brain Vault has been funded for the past many decades by primarily the National Institutes of Health. We've been writing grants to NIH, to NSF, to the Department of Defense, and most of these grants are in. Even if they are funded, and the likelihood, we don't even know if there will be an NIH. The likelihood of, we have to be considering other models for funding in general. We realize that as scientists, but also in our lab, we have this time where, the football season is going on. We want to continue to be testing these athletes. The concussion clinic is still going on at Lurie, and we want to continue to be working on these kids. So if anybody has ideas, mechanisms for how we can scale the work, and you want to learn more about what it is that we do, come and visit us. But we really are needing resources to be able to test the many, many athletes, do the many analyses that are necessary in order to hopefully bring this very, very powerful biological marker of concussion into the main, to make it the standard of clinical care. These are, so here is Sylvia, who I hadn't pointed, wasn't it before, and here's Trent Nickel, also very involved in this work, and then the other folks I've talked about in previous slides. And very, very importantly, I cannot let you go without telling you to please visit our magical website. If you click on any one of these things, you will get detailed information about each one of our studies. We study many things, and I encourage you to begin with the friendly overview slideshows and then go as deep as you want from there. Really, this website, and it's Jen Krisman who leads that effort, and actually Sebastian has been a big part in this effort as well in the last 2 years. It's a labor of love. It is there for, knowledge is empowering, and our little discoveries are there for you to have a look at. If you want to know more about our biological approach, there's a demonstration again on the homepage, a little video that's maybe 2 minutes long that you might want to have a look at, listen to as well. There's sound, of course. So I'm done.
So we have a few minutes for questions, if anyone has questions for Dr. Kraus.
Phenomenal work. Thank you so much for sharing it. This is all very new to me. I don't really have a background in audiology, so from what I learned, I have a question that might relate more to the clinical side of this. In looking at, you talked about strength of response, accuracy, and timing or processing of the response. can those be altered with any sort of environmental or contextual fact? Meaning, can they train themselves out of your test being able to identify?
That's such a beautiful question, because of course, yeah, we want to be able to assess the brain, but we also want to do something about it. And so, there have been a number of treatments for concussion. And what is, I think, very important is here you have this objective biological marker that you can use as a baseline measure in an individual who's had a concussion, and then very orderly and systematically look at the treatment, the impact of treatment on the brain measure and on the symptoms. So yes, this is absolutely something that I really see as one of the very important ingredients in having this biological marker, is to be able to assess different recovery strategies, because we need to be informed about what can we do about concussion, and being informed objectively, especially in a situation where taking behavioral tests has some limitations, I think is going to be important. Thank you for that. Questions? Yeah?
Looking at the FFR with the football players, do you notice any difference by position, so like a kicker versus a linebacker?
Yeah. So we've got all that data, and if we could stop writing grants all the time, I'm just saying, it makes us crazy that we have so much data already to look at, and data that we would like to be collecting to be able to really look at these questions. So we have information on every single football player in terms of their position, and what we need at this point really is numbers. We need to be able to track these guys longitudinally. We need to have more people in the concussion group so that we can actually look at questions like, what about position? What about history of previous concussions? What about they're coming in with certain language and learning problems? There are all kinds of factors that we want to look at that we just simply are underpowered to do with the current small data set. Yeah?
If the FFR has the potential to be a metric for recovery, marking recovery from a concussion, are there correlates in other neural pathways? Like, is there a correlate of the visual pathway of FFR or the somatosensory that you know of?
No, this is really, and I really tried to make the point that there's something, each one of our senses is special. And there's something very special about the processing of sound that enables us to have a window into that processing of sound through the FFR. And it is an auditory specific measure that relies on the very, very less than 1 millisecond speed of timing and integration that happens across neurons in the auditory system, which is not characteristic of some of the other sensory systems.
What, with the children at Lurie, what was the time between the incident where the concussion occurred and where the first data is collected? Because you said they were children with persistent symptoms.
Right. So do you have an average, what was it?
Probably about 3 weeks.
So so for the initial assessment,
Right.
we're talking an average of, 3 weeks later, these kids are still having symptoms, which is why they have gone to visit Cynthia.
So the hypothesis might be that if we could measure them
Earlier,
sooner,
yeah.
we'd see a more
Right.
depressed.
Absolutely, and that's what we're seeing in the football players.
Yeah.
Okay? And so what is interesting to think about is that with the Lurie kids, as you've pointed out, we're looking at the most severe concussion cases, right? So maybe this is 20% of 30% of people who have concussions continue to have symptoms that they are severe enough that they're going to see the doctor. With the football and the other athletes, we're looking at 90% of concussions that have symptoms immediately. And so the more common group, and again, as we learned, many symptoms resolve within this period. But here we have this measure to have a look at it. So yes, we have to be, and you know, again, you know, there cannot be better populations and also better populations to study together than we have at our fingertips. I mean, you know, we're able to look at immediate, you know, what is happening with immediate concussion, acute phase, versus longer-term recovery, and we're able to look at different times in the developmental lifespan, and you know, one or two things distinguish a 13-year-old and an 18-year-old. And so, you know, again, this is where being a biologist and being surrounded by people who know about language and sound processing and language development is really important.